DNA Replication and Protein Synthesis by Serena Vallieres. For my model, the key is white for the sugar molecule, black for the phosphate molecule, red for adenine, yellow for thymine, blue for cytosine, and green for guanine. This represents a cell. Here is the nucleus and here is the ribosome. In between the nucleus and the ribosome is the cytoplasm. DNA, deoxyribose acid, is located inside the nucleus. DNA has a sugar-phosphate backbone. In reality, it looks like this. The phosphate is on the outer side of the backbone, whilst the sugar is on the inside. A, T are complementary, and so are G, C. The lines represent the hydrogen bonds. You can see that AT has 2 and GC has 3. In my model, hydrogen bonds are represented by the magnets. DNA is a double helix structure in which it basically looks like a ladder but twisted. The process of DNA replication occurs when the DNA double helix unwinds. Each DNA molecule is a double-stranded helix. An enzyme called helicase unzips the two strands of the DNA like this. The helicase unwinding or unzipping the two strands creates a replication fork, leaving the leading strand and the lagging strand. The leading strand goes in the direction of 5' to 3', whilst the lagging strand goes in the direction of 3' to 5'. The leading strand is also known as the sense strand, whilst the lagging strand is also known as the antisense strand. Each separate strand of existing DNA molecule acts as a template for the production of a new strand of DNA. Nucleotides are picked up by the enzyme DNA polymerase. As DNA polymerase goes up the leading strand, it collects free nucleotides floating inside the nucleus and attaches it to the exposed ones, like so. As the enzyme goes up the exposed bases, it joins with the complementary bases, which creates a double helix DNA. The lagging strand is in the direction of 3' to 5', which makes it difficult with DNA replication, as the DNA polymerase can only go from 5' to 3'. This is where the enzyme RNA primase comes in. It goes and makes a starting point, and then the polymerase can work backwards from 5' to 3'. As you can see, the RNA primase initiated the base bonding, whilst the DNA polymerase followed on with the complementary bonding. This is called an Zaki fragment. This occurs over and over again until the whole lagging strand is replicated. Each resulting DNA molecule contains one strand of the existing DNA molecule and a newly synthesized strand. The replicated DNA molecules rewind into the double helix conformation, like the original molecule. The end result is that there are two molecules of DNA. Each are a double-stranded helix, and they are now identical to each other and the original molecule from which they are formed. Transcription. Protein synthesis begins with a gene. A gene is a section of the DNA molecule. The helicase unwinds or unzips the two strands, breaking the hydrogen bonds and exposing the bases. Next, DNA polymerase collects free nucleotides and connects them to the non-coding strand. In transcription, instead of replicating the strand, they are making mRNA. The difference between RNA and DNA is that this sugar is called a ribose sugar and this is called a deoxyribose, hence the name RNA and DNA. This is a single strand, and instead of the thymine you have a uracil as shown. As you can see, these are the same apart from this is T and this is U. Therefore, the mRNA looks very similar to the coding strand. So this is the coding strand. This is the end of transcription. Now the mRNA is able to leave the nucleus in which transcription occurs through pores and enter into the cytoplasm. The non-coding strand and the coding strand rejoin as a DNA. Translation. Once in the cytoplasm, the mRNA attaches itself to the ribosome. The ribosome is actually made up of RNA, which is ribonucleic acid. In the cytoplasm, the tRNA goes and searches for the amino acid which meets its sequence. tRNA has three bases, which is called a codon. The tRNA then delivers the amino acids. So you can see that U and A are complementary, so they join up. This then follows on with AU, AU, and AU. So this creates lysine. And as you can see, these two amino acids are joining. Histidine joins up to the mRNA also. And so does glycine. And finally, so does serine. This chain of amino acids is called a polypeptide. In reality, this chain of amino acids creates a protein. So this is delivered off and the tRNA leaves the mRNA in search for more amino acids. This chain is used for processes in our body. This is the end of translation. Thank you for watching my video. You just coughed. How do you feel about that?
This is the end of the worst biology assignment in history. Please advise people to use Play-Doh and make it much simpler in the future. Thank you very much for watching.